the second lecture for module 2, that is dealing with protein architecture, we will look at the secondary structure of proteins. We looked at the primary structure, which was the sequence of the amino acids, and from the sequence of the amino acids we found out that the connectivity, that is the peptide bond, is the covalent linkage that holds the sequence together, and in addition we had the disulfide bonds. In the secondary structure component, what we are going to understand is we are going to study what we mean by an alpha helix, a beta sheet, and construct a helical wheel and see what this means. In addition, the most important being the geometry of the backbone in the specific torsion angles, which we touched upon a bit in the last class when we also spoke about the rotamers for the amino acid residues, that is the rotation about the single bonds that are possible, and here we will discuss the important Ramachandran plot. The 3D conformation will come later when we consider the three-dimensional structure of proteins and how they are formed in protein folding. And we will look, as I mentioned, on helices sheets, the peptide backbone, and the important torsion angles, the foci angles. Now, what we mean by the secondary structure of proteins, we understand that the primary structure is the linking of the amino acid residues together through the peptide bonds, and we understood that this could form just a straight chain. But we know from our knowledge that it does not remain as a polypeptide chain in a straight form, it folds. Now, this folding can have specific patterns. One of these geometric patterns that is very unique is the formation of the secondary structures. This secondary structure can be what is known as the alpha helix or the beta sheet. What we see in this particular protein, that is ribonuclease A, this is a segment of the protein that forms a specific geometrical pattern. This is another specific form that is present in the protein known as the beta sheet, that is formed from separate strands, and these give rise to recurring structural proteins or patterns that are observed in proteins. We will look at these in a bit more detail, and another factor is the turns that actually are linkers to these secondary structure elements, as they are called. So, when we look at protein secondary structure, we look at a helix, the alpha helix. The most common alpha helix present in proteins are known as the right-handed helix, and the way we know that this is a right-handed helix is its direction of propagation. And if we consider our right hand and we consider the direction of propagation, we know that this is a right-handed helix. Now, there are specific characteristics to these helices, and there are reasons why they are held together in such a spiral fashion. This is because we have the main chain form this spiral form, and the connectivity is through specific, or the interactions is through these specific hydrogen bonds that can be seen. They are formed between specific amino acid residues, which we will see in a moment. When we look down the helix axis, so if this is the helix axis, if we look down the helix axis, this is what we will see. So, this is our helix axis, and what we see is we see surrounding the helix axis all the side chains protrude out from the helix axis. We will see how this is important in several aspects. The other secondary structural element is the beta sheet, that is formed of strands. These strands can be parallel in fashion, or they can also be anti-parallel in fashion. And in addition to this, when we look at the side chain residues, the connectivity in this case is also through hydrogen bonding interactions. So, this is what holds secondary structural elements together. So, the common feature of the alpha helix and the beta sheet is that they have hydrogen bonding interactions that hold the helix the way it is, or the strands, be it parallel or anti-parallel. Another type of helix that also can be formed is the 310 helix. We will look at these specific types of helices in a moment. So, when we look at the hydrogen bonding patterns of these helices, the common hydrogen bonding pattern in the one that we usually refer to as the alpha helix is the hydrogen bonding between the I and the I plus 4. So, we are looking at, we had studied what the hydrogen bonding pattern, so we are looking at a hydrogen bonding between the peptide backbone residues. So, this is what we are looking at. So, if we look at a specific type of residue, so this is our C double bond O and this is our NH behind there. This is a C double bond O and this is our NH. So, this bond is the hydrogen bond that holds the helix together in this particular type of orientation. So, when we have this I to I plus 4 bonding, we have the pattern. So, we have I to I plus 4, then I plus 1 will have a hydrogen bonding to I plus 5 and so on and so forth, that will hold this in this particular fashion where we will see the side chains protruding out. Now, this means that there is a particular turn of the helix and there are 3.6 residues per turn of the regular right-handed alpha helix that has the hydrogen bonding from I to I plus 4. This means that there is a pitch to the helix which is 5.4 angstroms in length. The hydrogen bonding distances are usually around 3.4 angstroms. So, if we look at the types of helical structures that are present in proteins, we mentioned the alpha helix, that is the common right-handed alpha helix that has the hydrogen bonding between the I and the I plus 4 residue. There is the 310 helix that has the H bonding between the I and the I plus 3rd residue, and then there is also a helix called the pi helix which has a hydrogen bonding from the I to I plus 5th residue. So, it means that if we look down the axis of each of these helices, we will see that this would have a tighter, because the rotation would be such that there would be a pitch that is smaller. Here we would have the I to I plus 4 and this would be a bit larger because we would have an I to I plus 5. Now, due to the hydrogen bonding pattern, there is also an overall dipole moment, because we know that there is a directionality to the hydrogen bond. The hydrogen bond, as we learnt, is formed between the C double bond O and the NH, the C double bond O of the ith residue to the NH of the I plus 4th, in this case I plus 3 or I plus 5, and that is how we have the hydrogen bonding pattern in the specific helices. Now, what we can do is we can plot what is called a helical wheel to visually represent the alpha helix, which we will see in a moment after our discussion on beta sheets. For the beta sheets, what we observe is the hydrogen bonding pattern again between the polypeptide chains. Now, what we observed in the hydrogen bonding pattern is a pattern that looks at a specific set where we have again the C double bond O hydrogen bonded to the NH. Now, when we look on the pattern that is shown on the left, this is a beta pleated sheet that has specific strands to it. What do we see? We see 3 of these strands that are parallel in nature. We see one that is anti-parallel in the sense that it is going in the opposite direction. Now, when we look at the hydrogen bonding pattern of these, we try and observe the directionality first of the backbone. We know that we have this particular direction of the polypeptide chain in this direction. Now, when we have the C alpha, we try to recognize the C alpha, saying that this is one particular amino acid. And how do we know that? We know that the C alpha is connected to the NH and the C double bond O. Similarly, if we look at the strand that is going in the opposite direction, this is also one amino acid. That says that this is the beginning of the amino acid, this is the C alpha, and this is the end of the amino acid because it is going in the opposite direction. Now, if we look at the hydrogen bonding pattern between these two specific strands, we will observe that this amino acid, the C double bond O, is connected through a hydrogen bond to the NH of this amino acid, and the C double bond O of the same amino acid is linked through hydrogen bonding through to the NH of that same amino acid on the other strand. This is unlike what we observe for the parallel case, where we will see that again if we look at the C alpha here and the C alpha here, what we observe is that the NH of this particular amino acid or the C double bond O of this particular amino acid is hydrogen bonded again to the same type of chemical moiety, but to different amino acids. So, this is the hydrogen bonding pattern difference that we observe for the parallel strands and for the antiparallel strands. For the beta pleated sheet, where we saw the hydrogen bonding pattern, we realized that this particular hydrogen bonding pattern is different for the parallel and the antiparallel sheets. And while we look at the hydrogen bonding pattern, we can distinctly see what kind of strands are forming this particular beta sheet. Now, interestingly, if we look at the direction and the pattern, we look at the hydrogen bonding. Now, how is this important? We realize that when we are looking at the two different types of secondary structures, that is the alpha helix and the beta sheet, the strands in the beta sheets, they are held together by hydrogen bonds. This is what is common for the alpha helix and the beta sheet. But what we realize in the beta sheet type of characteristic hydrogen bonding, the strands, whether they are parallel or anti-parallel, will have different patterns. Similarly, the hydrogen bonding nature for the alpha helix would depend upon the type of helix that we have in terms of whether it's going to be I plus 4, I plus 3 or I plus 5. Now, another major difference between the alpha helix and the beta sheet is their presence or their specific locations in the polypeptide chain. While in the formation of the alpha helix, we realize that we have to have a continuous chain. In the beta sheet case, the strands can come from different parts of the protein and they may not be continuous in nature, in the sense that we could have even a helix between the strands of a beta sheet. So, while we are looking at an alpha helix, we know that it has to be continuous in the polypeptide chain, but the strands in the beta sheet can come from different regions of the polypeptide chain, bringing them together in specific patterns that are known as the secondary structure beta sheet. What we will now look at is what we mean by a helical wheel. Now, the construction of a helical wheel is very simple and it is very interesting as to the information that it can provide us with. What we need for this is we need only the amino acid sequence. So, a knowledge of the amino acid sequence which is forming a helix is enough for a helical wheel construction. Now, what we know from looking down the axis of the helical wheel and understanding that the number of residues that we need for one complete turn for a regular alpha helix is 3.6. So, given that a complete circle is going to be 360 degrees, we know that when we go from one residue number 1 say to residue number 2, we are going through a rotation of 100 degrees and the rise is 1.5 angstroms. So, that this implies that for a complete turn of 360 degrees, given that we need 3.6 residues for a complete turn, for each residue the turn is 100 degrees and the rise is 1.5 angstroms. That when we multiply by 3.6 gives us the pitch equal to 5.4 angstroms. So now if we look at how, now look at the disposition of the side chains. They are all away from the helical axis. So what do we mean by this? We mean that when we are looking down the helix, what we see is if we have a specific residue and then we want to look at the location of the next residue in the helix, it is going to be 100 degrees away. So the representation can also actually predict which part of the helix is likely to be on the surface of the protein. When we look into more of protein folding, we will realize that for regular globular proteins that have a solvent like water around it, what happens is the hydrophobic residues tend to be in the center of the protein, which means that hydrophilic ones would be on the surface of the protein. So if we look at a specific helical construction, we will actually be able to predict in a manner which ones are likely to be at the surface, or which face of the helix rather is near the surface. So let us see how we can do this. So we look at the part of the helix that is likely to be on the surface of the protein. When we look now at an amphi, that means that we have an amphipathic helix in a sense that one part of it is going to be hydrophobic in nature and one part can be hydrophilic in nature. But then we can also ask the question is whether this is always true. May not be so. We will answer this question as we go along. So now say we have just an amino acid sequence, and what do we need? That's all the information we need to construct the helical wheel. What do we know? We know that residue number 1A is going to be at the top, say. The second residue serine is going to be 100 degrees. So if we just plot a circle like this or just draw an axis like this, we know that this is 90 degree. So we can say that this is around 100. So if alanine is here, it means serine is here. D, that's aspartic acid, is going to be 200 degrees. So we know that this is 180 degrees. So 200 is going to be somewhere around here. So the next amino acid will be here. So similarly, we can go on doing this construction till we cover the whole sequence of amino acid residues that forms the helix. What do we see? This is what we see, and this can be done very simply by using specific web or internet resources that are available to do this. So now let us look at the pattern. So this was the first amino acid, the alanine, then the serine, then we can see the aspartic acid and so on and so forth. Here is our F. Now if we look at the pattern of this, we see that the ones that are boxed are the ones that are hydrophobic in nature. We see some sort of a pattern in the sense that if we draw a dotted line along, say somewhere around here, this part is hydrophobic in nature and this part is relatively hydrophilic in nature. So what would we expect if this helix were part of a protein? We would expect this region to be near the center or near the core of the protein and this region to be on the surface of the protein. So this is a very interesting way just from the sequence to identify which part of the helix is going to be present where in the protein. Our next important aspect of understanding of the geometry of the backbone, which we touched upon in the previous class for the primary structures, is an understanding about the rotation about the single bonds, because when we look at the rotation, we are looking at specific geometry, and when we look at the specific geometry, we can try and correlate this with the pattern that is observed in the strands and in the alpha helix. And this was done by Professor G. N. Ramachandran, which is now known as the Ramachandran plot, an extremely important plot in protein structure that decides upon some specific allowed regions for proteins and allowed angles for proteins. We will see that what we mean by the allowed part. So, here is our psi angle, here is our, it is the torsion around the C alpha C bond, as you can see here. The phi angle is the torsion around the N C alpha bond. So, when we are looking at the N C alpha C O, this is what our backbone is. You have to remember the backbone. The backbone is N C alpha C double bond O. So, this we do not consider the H or the R for this right now, but this is how we would actually, the orientation is not particularly correct, but this is how we would consider our C double bond. So, the N C alpha C O, and so this would mean that this comes, the next N comes here, right. So, now when we have the rotation about the C alpha C bond, that is the psi angle. When we have the rotation about the N C alpha bond, this is the phi angle, this is the psi angle. What is this? This is our peptide bond. Our peptide bond rotation is also possible, known as the omega angle. So, we have the omega angle, the phi angle and the psi angle. So, the omega angle that we see is for the peptide bond. It is trans when it is 180. We know what the trans peptide bond means. We know what the cis peptide bond means, meaning an omega angle of 0, where we have the C alphas pointing in the same direction. The Ramachandran plot is now a visual description of the combination of the phi and the psi angles that are permitted in a peptide backbone, but are not permitted due to specific steric constraints. And interestingly, the helices are in this region, the beta sheets are in this region, meaning that their phi psi angles, because of the structural constraints, because of the specific geometrical patterns, occupy specific regions of the Ramachandran plot, and some of these regions are not allowed. Why are they not allowed? These are the allowed values, and when we look at the allowed conformations, there are possible peptide conformations that will not have steric interference. We do not want this rotation about the single bond to result in a clash, and the conformation which both phi and psi are 0 is not possible because of steric interference between the atoms in the polypeptide backbone. What is this? So, when we have the rotation, they cannot rotate. We know that the peptide bond is planar. Why? Because it has that partial double bond character, but there is a possibility of phi and psi rotations. So, if this has a partial double bond character and is not allowed to rotate by itself in the omega angle, the phi and the psi rotations are possible. But again, when we look at the specific C alpha attachments to, or rather the side chain attachments, we realize that when that bond is moving, there is a possible of steric clashes. So, now when we have the movement about the phi or the psi angles, we see the clash. Now, with that clash, what comes is impossible or phi angles that cannot be allowed due to the steric interactions. So, the phi psi interactions again give us specific regions of the Ramachandran plot that can be occupied, where clashes will occur for other possibilities. This is a very important feature of the backbone of the protein structures, and this allows only specific orientations to be possible. So again, if we have we can rotate about the psi angle and see the same distinct possibility where we see other regions that form clashes and some regions that are allowed, and these regions will be allowed for the beta and these regions will be allowed for the alpha helix, the important secondary structural elements of proteins. So what we covered was the secondary structure, the alpha helix, the beta sheet and the helical wheel construction, the specific torsion angles, and most importantly the Ramachandran plot. Thank you.